Awesome. So let's get started. Thanks everyone who's tuned in today to hear Ratul talk about COVID and help us get more informed. As the restrictions are starting to ease up a little bit, we all thought here at the Popuri project that it might be a good way to familiarize ourselves with what we're dealing with and what the future looks like moving forward, living with COVID and what kind of cures we're looking into. So yeah, over to you, Ratul. Let's talk more about you.
Yeah, so I'm good. I'm good. Thanks for inviting me for this. It's a
Looks
great pleasure.
like we have a video frozen.
Did the video freeze?
Yeah, we're good now. Go ahead.
All right. So yeah, I mean, thanks for getting me here. I like talking about science, and I think today I'm trying to give like a layman's explanation, so that like even if someone's not familiar with bio, they don't have they at least take something home. Because I mean, it's just that every discipline has a lot of jargons that takes it away from the general crowd, and
Mhm.
I do not really believe in that. So what I'll do is try to keep it as simple as possible. Obviously, we have to get into some bio at some point to explain what's going on, but
Right.
I'll build up a framework which is non-bio, so that you can analogously map in your head as to like, okay, this means that, hopefully. But if you have any questions, please put it in the comments. And I think Anupriya and team are doing a great job at this, like managing everything together. So yeah, I
Uh,
mean, if you have questions, just post it. If you don't see it live, we'll address it whenever we get it.
Sounds like a plan. Awesome. So let's get into it.
Okay, so I go ahead with it.
Yeah,
Okay, so I will share the screen and use my slides to basically explain what's going on. One sec, let me see. Okay, so one sec. One sec. Sorry, this can get messy at times.
that's okay. We're all dealing with multiple screens and screen shares all this while, especially due to lockdowns and stuff, right?
Yeah. One sec. I think the best way to go ahead with this will be. Okay. So do you see my screen?
Yes, I do.
But
Uh,
do you see it still?
yes, I do. Now I see
Okay.
it full screen. Yeah.
Okay, perfect. I was just hoping that this
Awesome.
works out. Okay, cool. So let's get started.
Check
So
out. Yeah.
yeah, I'll be actually dividing the whole talk into like three parts. First, giving out the basics of what proteins are, and so that like proteins are the heart of COVID-19 and what's going on. So it's better to give an explanation as to what proteins are, how they're formed, and how they function, etc. So I'm trying to use a bit of analogy here to explain what's going on. So let's go ahead with this. See, we have a quarter, let's say, and there's a machine which just accepts quarters. So we have quarters only. So and let's say this the lower slot of this will give you out $1 for every 4 quarters that you put in. So essentially, if you put in this 4 quarters through the upper slot, you what you get is the equivalent amount, but it's in the form of a note. So it's a converter. So now, for the sake of this talk, let's imagine that there are four colors of quarters. So we have like white, blue, green, and yellow. This is just hypothetical. So we'll code each of them with the single letter alphabet, starting the first alphabet of the word. So WBGY. So let's imagine a situation where there's a machine which will actually give you the color of a note depending on what sequence and what colors of coins you've put in. So let's say these coins will be read in the direction WGBY. So basically, first W goes, then G, then B, then W. So WGBY corresponds to a red note. So we can say that when we feed WGBY in this order, we get R as an output. So similarly, let's say that if we use an alternate sequence of these four coins, you get a different color of note. So now there can be several multiples of combinations of these. So based on what input code you feed, you get an output code. Let's imagine this way. And for the context, let's imagine that there are only 20 feasible such outcomes. I'm sure combinatorially there are many other outcomes, but let's say we will connect this to biology at some point. But we are saying that let's restrict the total number of outcomes to only 20, and we know which 4-letter code corresponds to which 1-letter code on the right. So now imagine, instead of just feeding discretely 4 coins, you just have a huge string which has 4 coins, 4 coins, 4 coins strung up together. So we have a multiple of the number of coins is a multiple of 4, and you feed them together using an automated machine where as soon as 4 of them go inside, one note comes out, and the next frame goes in, and the another note comes out, and the next frame. So that's how you will generate a sequence of notes, and the number of notes you'll notice will be 1/4 of the total number of coins fed, because 4 coins correspond to one note basically. So, but now trying to move towards biology, what's the analogy here? Why have I been saying all this? So let's imagine that that this machine that we had is a converter. We call this biological machinery called ribosome. So, and ribosome does not take in coins, but what it takes in is an RNA sequence. And for people who know, it's a messenger RNA sequence. So yeah, and it is read the same way that I explained. So the sequence of this RNA just goes inside the ribosome, and what comes out is obviously not notes. What comes out is a single amino acid. And based on, and these are not 4 of them analyzed together, there are 3 of them which correspond to one single amino acid. And the next 3 are being read, and the next amino acid comes out. Next 3 are being read, and the next amino acid comes out. And so, in a way, you can imagine, as this whole RNA sequence is being read, a sequence of amino acids comes out, and they look like like beaded a string of beaded beads basically. And this string of beads is actually called a protein, and those individual beads are called amino acids. So, so essentially, if you represent each of the amino acid with their single one-letter code, the necklace or the beaded structure would essentially look something like this. And but this is not how a protein looks like. This is way stringy and non-functional. It just is like a thread, if you can imagine, only in nanoscopic scales. But what happens is that they undergo a process of complex folding, which looks something like this. You have this beaded structure that folds upon itself and upon itself and upon itself to ultimately give you this finally complex quaternary structure, which ultimately is known as a functional protein. This is what a protein would look like. And for the sake of argument, I'm going to explain today that the proteins are classically represented in 3 different representations. One is called the cartoon representation, and the equivalent solid sphere representation. I think the middle one is something that we are very familiar with since our like chemistry in high school, where like you have individual ball and stick models of each and every atom that exist. And the final structure, which is the surface representation, where you just smoothen a surface on top of what is being represented here. This is what basically the classical representation which I'm going to use for the rest of my talk for all proteins at hand. So we'll be looking at this kind of a surface, which is ultimately comprised of atoms in the at the very core of it. So so the interesting thing about COVID is, so COVID will definitely be known as the 2020 pandemic. And why is this a great havoc? Is that so the weight of a single virus is roughly 10 to the negative 14th grams, and each infected individual has around 5 10 to the 7th number of such particles inside their body, and the total number of infected as of today is 5.47 million. So if you multiply all of them, all you'll get is that there's 2.7 grams of virus that's creating all of this havoc worldwide. And this is this is essentially adds up to the weights of 2 M&Ms, and the M&Ms are drawn to scale, by the way. So you can imagine. So we really need to get to the deep of it as to what's going at the microscopic level. That 2.7 grams is creating what it has created today. So this is a picture I think you have seen all over news every day, and like and various social media outlets, etc. This, yes, this is the coronavirus, and the red inverted triangular structure that you see, there are multiple of them which protrude outwards. This is called the spike protein, and if you look at the structure, the spike protein is an inverted triangle, but it is itself composed of 3 identical subunits which come together in a pyramidal fashion and to form something like this. So you have the second unit and you have the third unit. Together, they constitute this spike protein. Now, for the sake of clarity, I am going to color each of these subunits in 3 different colors, which will look something like this. And if you rotate this, this is how the 3D structure of this spike protein, which is embedded on top of the virus, looks like. Now what's going on? So you have the virus. If you inhale it, or like somehow ingest it through your respiratory or food tract, it goes in and interacts with the lower throat, which is also known as the upper respiratory tract. And this is a microscopic image on the right of the lining of the respiratory tract. And this virus particle actually is goes through and sticks somewhere here. So what do I mean by sticks somewhere here? So if you look closely, what will happen is, what you'll see is that there are such some proteins which are which are surface exposed, which means this is where the virus comes from. They're exposed on the surface. There are many of them which are on human cells. These protein, for the sake of simplicity, we'll call this protein H, because H for human. This is a human protein which is at the surface of all human cells, most human cells, let's say. So now let's say on the left you see a big image of this virus, and this is one such spike, and this is another spike. So what happens is, and when it comes close to this H protein of the human respiratory lining, it basically flips open like the flap of an envelope, as if it's reaching for its friend, sort of. And because they are oppositely charged, they kind of pull each other, and there is like a strong attraction between them. So it pulls towards it. The virus basically gets attracted towards this protein, and then the whole thing is internalized into the cell. That's how the virus enters your body. So this is the step zero of virus infection. So the point is, what are we trying to do in terms of medicine? So the main objective here is to destabilize this kind of a structure, or prevent this from happening at all. So how are medicines expected to work? We see that people are doing a lot of drug-based drug discovery approaches, trying to figure out what's the right drug. We see that this drug was tested, but what they're essentially trying to do is that they're trying to figure out one drug which will ultimately find its way and sort of destabilize this complex and sort of break this. And what will happen if it breaks this? The virus that makes an attachment with this protein on the human cell surface, that will be completely broken. And as long as the drug is there, which means as long as you're taking the drug daily till you're cured, this virus is not able to enter your body. So but the search for this drug is still on. So that would prevent viral entry. It's not just this phenomena that the drug has to control. There are several other downstream processes. It should not create an imbalance in your body, etc. But I'm not going to the details of that. But when I'm talking about drugs, one very interesting thing that I must mention is that the patients who suffer from high BP, they take some sort of a drug which essentially does not do this, but creates more of this yellow. So creating more of this yellow means there are more such elements to bind with the spike. So that's why people are saying that people who are on heart medications, they might be susceptible to the virus, not because of immunity, just because their medicine creates more of this yellow protein, which likes the virus and tries to pull it in. So there's a second class of heart medications, which is basically called ARB blockers. So those are the ones that are recommended because they do not have this side effect of producing more of this yellow protein. I think that's what is being recommended right now. So and the other the, so this was a small digression, but again, so this is drug is one approach by which you can prevent this from happening. And the second way people are trying to do is trying to get some sort of an antibody. Antibodies are defense molecules which are produced by one's own body to fight any incoming disease protein. But what if you do not have it? Then if you do not have something, then you definitely fall sick. Then you need to take that particular antibody as a vaccine, or through a syringe directly injected into your bloodstream, so that it can now show up on your cell surface and prevent this protein from actually approaching this yellow protein, which is the human protein which likes the virus and takes that in. So this gray barrier is like a Y-like structure. We'll make a lot of them on the surface of it. If you inject it into someone's bloodstream, they will position themselves appropriately on the cell surface and prevent this virus from coming in. So this is definitely called antibodies, and this prevents denies the access basically. So the question right now is that how dangerous is the virus in comparison to other coronaviruses? Because we already know that this one's a pandemic, whereas I think the previous coronavirus outbreak was not half dangerous. So some of our preliminary calculations actually show that if this is a scale, this is like an arbitrary scale which represents infectivity, like how easily humans are infected by a certain strain of coronavirus. This is some other strain, which is this is a intermediate strain, and this is like coronavirus 1, which happened in 2002, and you have coronavirus 2 here. Our calculations sort of indicate that like coronavirus 2 is 45% more infective, which means human cells tend to like SARS-CoV-2 virus more by 45% than SARS-CoV-1. So the propensity to fall ill is much higher. So the question is, so how worse can it get? So now this is a little bit slippery slope. So basically what can happen is that since we know how the virus can interact with the human cell, and since we know what's the strength of its attachment, it is possible to genetically manipulate the virus in a way that this strength is further increased. And our calculations sort of indicate that you can enhance this effect by up to like 25%. The strength can go up more than 25%, which essentially means that we can have an even more potent SARS coronavirus, which is scary. But the good thing is that it could be contained because we now know what are the changes that needs to be introduced in this interface so that the binding will be improved. Since we already know what are the possibilities to improve it further, we can already have a medicine or like a drug that will counteract it a priori. So even if the virus becomes stronger, we should not have to worry about like a second outbreak which becomes like as massive as this, because I'm guessing there are like labs right now who are figuring this out. This process is somehow called a viral escape. It means that you make a drug to stop the virus from entering, and the virus tries to change itself to escape that trap rather, and try to get into the human body. So I right now, as far as I understand, the all the escape routes of the virus have been tracked down. So we are trying to make sure we can create one single like dose of medicine or sort of an antibody or drug which will contain this mutated forms of virus or more lethal forms of virus to still not enter. So I hope this was a little this could shed some light onto what's going on, and how much ever I understand. I'm clearly not the greatest expert in this, but this is my little understanding that I have. I think that's the end of it. I'll be happy to take any questions.
We had a few questions that came up, including my own. So let's
Okay,
start with the stuff that I've been thinking about while you were talking. So, based on the information that you have and how long it takes for a drug trial to go through until you have an achievable result which shows results,
right.
um, how far do you think an actual cure is
Oh, so
realistically?
no, so the thing is, there was a recent update from the Harvard Medical School. There's people have actually tested it on it in a certain version of monkeys, which closely resemble humans in their immune system, like the way to fight combat diseases. So it's like an exact model, so you could trust it. So the results sort of indicate that if you have it, the question is if you have it, then you will definitely get an immunity within 4 or 5 days, and once you have that immunity, that's for life. You can just roam around in the street without worrying anything. You don't have
I
to
see.
worry about whoever has it, whether the crowd is more than 5 people or not. That's it. But the thing is that they are also trying to make medicines on this. The thing is that the earliest estimate is that June 2021. So
Mm.
that's probably the earliest you can have a medicine which is commercially available. That's there. But also one good thing in this aspect that came out in the study is that the surface based contamination is now completely debunked. Surface based contamination, so wearing
I
gloves
see.
is okay, but that doesn't stop anything, because like it's not coming through surfaces. If you hold an Amazon package,
Right.
that's not affecting you. So question is, can you shake hands with someone? No, because you're in proximity with someone. That's the point.
Right, I see. And so another thing that I was thinking about was, does taking the medication, like you implied, if you're developing immunity to it is one thing, right? But when you're taking the medication, does that help you in the future as well? Like, does it make you immune in some way, or is it just
Yeah.
working on that binding link?
Yeah. So basically, if you take the antibody based stuff, like so the drug is not necessarily, so the drug works on a different level and the antibody works on a different level. The drug prevents the virus from entering. It does not give
Right.
you any immunity. It's just that
Right.
whenever this season comes, you'll probably have like a flu shot, you'll have a corona shot.
Right.
That's the thing. If that's the drug, but if it's a
Hmm.
vaccine shot, then it's giving you immunity forever. Then you don't have to take any more medicines. It's like that antibody is now yours. It's in your bag now. Every time the
I
same
see.
same attacker comes, you just throw it at it. So that's it.
I see. And so the preventative medicines that you were talking about, where they
Yeah.
would try to potentially work on what kind of mutations we might see further, might
Right.
also come in handy at that point. I see.
Yeah.
So another question we had was, how does the currently available medicines like hydroxychloroquine work against COVID? Is it like a similar concept?
Ye- yeah. So the concept is exactly the same as I said, as I showed that particular. You want to stop that interaction from happening with the yellow human protein and the blue virus protein. That's what you want to attack. So what people are doing is that trying to see what is that desired medicine that will prevent that complex from happening. The HCQ has been tested for that, so it did show some positive results. Because the thing is that this field has suddenly taken gotten so much attention. Like
Right.
thousands of papers are being written in a week, and I'm not exaggerating. So and
Hmm.
it's difficult to track all the information that's coming around, and there unpublished data is also being released for people to read, and that's also getting shared through social media, and sometimes things get become viral. So
I see.
HCQ, according to me, was one such case where maybe it showed some good effects to somebody. So HCQ will work if and only if it's in the very incipient stages and you are still in asymptomatic phase. If you take
I see.
HCQ, you're good. But the moment you're seeing symptoms, that it's beyond the purview of HCQ.
I see. I
Yeah.
see. That makes sense. So another question we had come in is it quite possible for the current strain to be 25% more potent?
Yes, that's what this is, a theoretical limit. Like that's what it can go up to. Question is whether it will go up to, to bring about changes within the virus. So the virus has to optimize a lot of things inside its body. So that,
Right.
even though theoretically for this particular binding this might be feasible, but the virus does not just do this, it has other bodily functions to carry out. So whether
Right.
that will be detrimental to its other bodily functions, that is what will determine whether the 25% can be ultimately realized or not by the virus. But even if it is, I think the current drugs are taking care of those roots.
I see.
Yeah.
Another question we had was, is it possible that a fetus gets infected? So if the
A
mom
fee
contracts,
Okay, so the fetus can a fetus gets infected. So as far as I understand, so the mom can transmit immunity, which is like these
I
antibodies,
see.
through the placenta. So if
Hmm.
the mom is immune, I do not think why the child should fall sick to it. But again, like the fetus getting infected means the virus has to travel through that respirator through the reproductive tract, which is unlikely according to me.
Right.
But again, like today what I say can become false tomorrow, because some new
They're
result
still
might come
figuring
out.
it out.
Yeah.
Yeah.
So I think, as far as I have read and as far as I know, the fetuses should not get it. But if there is an antibody in the mom, which is preventing the mom from contracting it in the first place, then the fetus should be safe.
I see. But what if the mom has contracted COVID? Does that
Yeah.
increase chances of contamination then?
So for that, I'm not sure whether the virus particle going down the throat of the mom will ultimately get to the fetus. So
Right.
that needs to be explored, because people I think that it's entering the body through the respiratory tract. If it's inside, then it's not going anywhere down, just entering your body through that.
Right.
But and if it's in the bloodstream, there is a possibility it might get there. Why not?
Right.
But yeah, this is still up for debate. I don't know.
I see. So Akriti asks, should we be socially distant until June 2021? Does that just make sense?
I think socially distant is just a way of life now. So
Yeah.
I think that makes sense. That's just pragmatic, and like just make sure. I mean, I think even if you're mixing with people, even if you're mixing with 4 or 5 people regularly every week, just make sure that they are not meeting other people, and you know what their schedules look like. I mean, you should be able to trust them to the point that what they're doing, where they're going. If they're going out multiple times to like several groceries and stuff, then probably you avoid.
Right. Um, Already recovered people are getting infected again. Could you please shed some light on that?
Right. So yes, so that's the only thing that I've read also, that previously infected people are also getting it, which kind of does not corroborate with what I am saying, or like what the recent study was. So I think they are, to understand why this is happening again. So either you need to sequence, like figure out the sequence or like the structure of this new virus that's coming out. So for that you need
Mm,
human testing, like to figure out
right.
what's going on, or you need an appropriate like monkey based model to again reinfect it and see like that. And the recent study that I saw, which explains what's going on, they see that there is no coming back of the disease second time. So but there's a recent report which I also saw. People might get it second time, they might be detected positive, but they will be asymptomatic. You will not feel it.
I see.
Your body will be fine, just that you'll detect positive. That's it. It's
I
like,
see.
yeah,
So you can still be a carrier. I see. Okay.
you'll be a carrier essentially. Even if you get it the second time, you'll be mostly a carrier. You will not be like in the bed again.
Right. I see. Someone asks, is there a peer-reviewed paper for remdesivir expected by end of May? That's very technical.
Remdesivir, I think there are peer reviewed papers you're saying. So peer reviewed papers, I think there are a lot of, someone who's asking, I think they understand what preprints are, like before peer review already publicly release it. So there are a lot of preprints, yes. Whether you can trust them, yes, maybe if you look up who the authors are, whether they're from credible
Mhm.
institutes, more probably that's one way of going about it. But I think nothing has come to my notice that's like purely peer reviewed and has remdesivir in the title and like says that remdesivir does this. But there may be something. It's not like I've read everything. So I'm not sure, but that
Yeah, and
seems
like you
to,
said,
yeah, that seems to
go
show
on,
a bit
sir.
of promise, I think. Yeah, that drug. Yeah.
Yeah, and like you say, there's like new information coming out every day, right?
Yeah,
Yeah.
yeah, yeah.
So Akriti asks, personal question, what's your postdoc research about?
Oh, okay. That's a good question. I wasn't expecting that. So you see this complicated geometric structures, like looks like blobs and stuff. This is the structure of a protein. So I told you at the beginning that there's an amino acid sequence, like the beaded string, which ultimately gives you the structure. So right now it's still an open problem in research, that given the sequence of alphabets which constitute that protein, what the structure would look like. So that's an
I
open
see.
problem. I am trying to do something, I would say, interesting. So the interesting thing is that we are treating the amino acid sequence as, let's say, a language A, which is English, let's say, and the language B is the structure, which is defined by the angles, like which how things are bent and stuff like that. So let that composite matrix of angles be language B.
Mhm.
So we are using something like a Google translator, which uses like English to Spanish conversion. We're trying to use exactly that machinery to figure out how we can convert language A from language B, where one is sequence, the other is structure. And once we
I
can
see.
get that, what we will know is that we'll know the grammar of how the protein folds. If we know the grammar of how protein folds, then we can design any sequence, any structure we want. Let's say we want A to B reaction to happen. Okay, design a protein for me. That will do it. So then we should be able to do that using those grammar. But this is like a long-term goal.
That's awesome. That sounds really cool. How can computer scientists help in COVID research?
I think computer scientists should come to the forefront right now, and think that it's past grade 12 where you had to choose between computer and bio. Right now, it's the time to combine. So yeah, I mean, computer scientists are, I think, I should not generalize, but again, I think computer scientists are essentially better coders, and this is the right time to basically engage and try to apply for various bio heavy positions where you'll be just heavily coding away and analyzing the data and all of that. I think biologists are good at looking at the data, looking at an analysis, and figuring out whether this makes sense biologically or not. But they are not necessarily the best in terms of coding it up in the most sophisticated best way. So I think this kind of positions should open up a lot more, both in terms of academia as well as industry, where purely computer science, completely bioagnostic people will be required in more and more in bio.
Right. Especially for like building tools like sequencers and understanding all
Yeah,
of
why
that
not?
data, right? Yeah, makes
Surely, yeah.
sense. Do you have any credible open sources for COVID-19 data that we can look at or use as computer scientists?
Yes, there is. I can. There's a link basically. I can put the link. Where do I put the link after the talk?
We can
Maybe.
definitely share it once we summarize this talk. We can share it on our page.
Perfect, perfect. I have
Awesome.
something that actually compiles all the cases and stuff. If that's what he's talking about, the cases based stuff. Like if you want to do any sort of epidemiology, like how this is working in different countries, etc. Just the raw data. There's a lot of data there. Yep.
Awesome. If we haven't been able to make a virus for existing diseases like HIV, what makes us so sure that we will for corona? I believe maybe antibodies is what they're going for, or even like a
Uh,
drug, I suppose.
no, so the thing with HIV is HIV has a lot of modalities means that if you stop it from entering this way, it enters the other way. But and it can enter through receptors in a I think the process is a bit more complicated. I am not a HIV expert, but as far as I understand, the in HIV reduces the immunity and it hijacks the immune system. So does this. But HIV seems to be more potent and more lethal. And I think with coronavirus, the hope is that I think we have figured out already what the exact mechanism of entry is,
Mm,
which is very important to know the way it enters the human cells. So you're already at a point where you understand the physics and the biology of it, how it's entering and whatnot. And I think for what's that called, for HIV, even if you figure out one way, it escapes in various multiple ways. So we have not seen any escape studies with SARS-CoV-2, which says that this escape tracking is very, very difficult.
I see. So
Mhm.
had it been the case that it had multiple ways of entering the body, it would have been much more complicated.
Yeah, it could enter in one way, but when you stop that, it has multiple other ways. So I think if that number is less, it's more tractable sort of.
Makes sense. I see. I think that is the end of it for now, unless I see any more questions come up. But yeah, this was a lot of fun. Thank you so much for
All
coming
right.
on today for us. It was a lovely session. And all of you watching, in case you want to send this to someone, it's going to be live for a little bit longer because there's a lag, but it's also going to be on our YouTube channel. And we'll be sure to post the resources that Ratul mentioned on our page as well. So you can reach out to him if you have questions on his personal DM, and we'll link his Instagram profile there as well. But yeah, thank you so much for coming on
Thank you.
today.
This is my
Yeah,
pleasure. Yeah.
it was lovely. Awesome.
All right.
I think we're good to go. Yeah,
Okay. Cool.
awesome.
Are we still live?